It's been years since I've seen mitosis. Everywhere I go, I see mitosis on the walls, on the streets, on people everywhere.
Ali! I will show you the secrets of mitosis.
Thank you.
The purpose of mitosis is for cell reproduction. One cell is able to make two exact copies of itself. For example, if you fall down and scrape your skin, you're able to heal because the skin cells replicate themselves into new identical skin cells.
Interphase is the phase of the cell cycle in which the cell prepares for mitosis. The first step of interphase is G1. During this phase, the cell expands in order to ready itself for DNA replication and cell division. Centrosomes are replicated as well. This phase acts as a checkpoint for the cell to make sure that it is ready for cell division. If the cell is not ready, then it will enter a dormant phase called G0. Until the cell has expanded and can support replication, the cell will remain in this phase indefinitely. The cell has specific stages where the cell must assess its internal structure in order to decide whether it can divide or not. The first step is the G1 checkpoint, where a cell must check whether it is committed to cell division. Some of the factors that the cell will check are size, nutrients, molecular signals, mechanical signals, and the condition of DNA. If all of these aspects are in good condition, then it will enter the S phase, where the cell becomes committed to division. If not, then it will enter dormancy, also known as G0. Cyclins are proteins that control the progression of cells through the cell cycle by activating cyclin-dependent kinase enzymes, otherwise known as CDK. The first cyclin is G1 cyclin. G1 cyclins bind itself to CDK proteins during the G1 phase. When the cell has reached a certain size in the environment, the cyclins begin to degrade, which deactivates the CDK and leads to the next phase. If the cell has met the necessary conditions for G1, then it will continue to the S phase. In this phase, the cell begins to replicate its chromosomes until it contains twice its normal number. The cell will also continue to increase its size. If a cell does not copy DNA chromosomes during the S subphase, the S phase cyclin will not activate due to the CDK, preventing the cell from moving on to the G2 phase. The cell will remain in the S phase until it has copied its chromosomes. If this can't be fixed, it will enter into a programmed death. It is important that the requirements for each checkpoint are met. If these checkpoints are bypassed, this can result in uncontrolled growth, which can cause diseases such as cancer. S-phase cyclins regulate progression through the cell cycle during DNA replication. Once the cell has correctly copied its chromosomes, the cyclins will be activated by CDK, allowing it to move on to the next step. The next checkpoint is G2. The primary concern of the cell is the condition of its DNA. The two main things the cell will check are DNA integrity and whether the DNA was completely copied. If there is something wrong, the cell will pause at this checkpoint in order to fix itself. If the damage is irreparable, the cell will enter into a programmed death, also known as apoptosis. The G2 phase is comparable to the G1 phase, as it acts as a checkpoint for the cell to make sure that it is ready for the cell cycle. The cell can check to make sure if it has properly duplicated its components, such as the chromosomes, for example. If the cell still needs to increase its size, then the G2 phase presents a final opportunity to do so. The final checkpoint is the M phase, where the goal of the cell is to evenly split its chromosomes between the two daughter cells. The two main things the cell will check here are the alignment and tension of each chromosome. The M cyclin, otherwise known as the mitotic cyclin, is responsible for deciding whether the G2 cell can enter into mitosis. When M cyclins bind to CDKs, this results in the creation of the MPF, or mitosis promoting factor. This acts as a signal for the G2 cell to enter into mitosis.
During prophase, the two chromatids that are genetically identical pair up with one another and condense into egg-shaped structures called chromosomes, which are visible. Centrioles and spindle fibers also begin to form. The centrioles and spindle fibers extend across the cell and move onto opposite sides. The nuclear membrane dissolves, which releases the chromosome. In metaphase, the chromosomes line up neatly at the equator of the cell. The spindle fibers from the centrioles, which are at the opposite ends of the cell, attach to each of the sister chromatids. If the pairs of chromosomes don't line up correctly, the structure of the chromosomes can be altered and mutations may occur, since the resulting daughter cells will have an abnormal amount of chromosomes. This can cause genetic disorders. In anaphase, the sister chromatids are pulled apart by these spindle fibers. which results in one chromatid at one end of the pole and another chromatid at the opposite end of the pole. If the chromosomes fail to separate, it causes non-disjunction, where sister chromatids fail to separate at anaphase, causing a genetic disorder such as Down syndrome. During telophase, at each end of the pole, the chromosomes gather together and two nuclear membranes are formed at each end of the cell. The spindle fibers also disappear. Finally, in cytokinesis, the cell pinches in the middle to separate into two new daughter cells, each with the identical copy of DNA, a full set of chromosomes in the nucleus, and the same organelles as the original cell. Cytokinesis in a plant cell. Plant cells have a cell wall, so Golgi vesicles develop in the equatorial plane and fuse together until a plate is formed. The cell plate is formed during telophase. The cell plate joins the plasma membrane and later turns into the cell wall, which makes two daughter cells. In animal cells, a cleavage is formed in the middle of the cell. This cleavage is formed and controlled by cytoskeleton elements composed of actin and myosin, which are responsible for pulling the plasma membrane inwards, the pinch. This then creates two new daughter cells.
After learning about mitosis, everywhere I go, I'm not scared anymore. Everywhere I look, I feel enlightened by everything I see. Mitosis has been conquered.